So today we are going to cover heme and hemoglobin, that is pigment metabolism. And in this, the first topic that is going to be covered today is basically the heme metabolism, the synthesis of heme, the regulation of heme, and along with that we are also going to cover the topic shunt bilirubin for additional knowledge, okay, about bilirubin. Now, what is basically RBC? RBC is nothing but red blood cells which are present in our body. They are biconcave in shape. Apart from their shape, their diameter is 7 microns. RBCs contain hemoglobin, and this hemoglobin is made up of two parts, heme and globin. Apart from that, RBCs which are there, they are synthesized with the help of erythropoiesis. RBCs are containing red color pigment, which is hemoglobin. That's why we are going to study pigment metabolism or heme metabolism. First of all, heme which is there, it is the two parts of hemoglobin, right? Heme and globin. Globin is said to be the protein part, while heme is said to be the prosthetic group. Okay, protein part and the prosthetic group. Heme which is there, the prosthetic group present, it basically has certain functions. The functions of heme first include the transport of gases which are transported. Apart from that, the second function of heme is said to be electron transfer. In case of citric acid cycle, there is transfer of electrons seen with the help of heme. The third important function of heme is said to be catalysis. Okay, catalysis. And the fourth function is antioxidant defense. And the fifth function, which stands for signal transmission, or transduction is also used. Okay, both mean the same, transmission or transduction. That is, if there are two cells, transmission of signal from one cell to another is done with the help of the heme group. Now, apart from that, where all heme is present? Heme is present in hemoglobin, it is present in myoglobin, it is also present in enzymes. Okay, these enzymes include catalase, peroxidase, nitric oxide synthase. Apart from that, there are also cytochromes which contain heme. Example is cytochrome P450. Okay, which is present in the liver. Now, if we move ahead, we'll see the structure of heme. The structure of heme which is there, it is said to be a derivative of, or we can say it is derived from, porphyrin. Porphyrin contains 4 pyrrole rings. These pyrrole rings which are there, along with Fe2 plus in its +2 state, okay, iron in its +2 state, and 4 pyrrole ring together form the porphyrin, which forms the heme. So this is pyrrole ring. If we make 4 of them, we would get porphyrin. If we add iron to porphyrin, we get heme. Okay, simple as that. Now, how is the structure of pyrrole ring present? Pyrrole ring basically is consisting of a nitrogen group over here, NH group, and there is presence of double bond on both sides. So this is the structure of pyrrole ring. Now, how is porphyrin present? Pyrrole ring. What happens is the 4 pyrrole rings should be connected to each other, so they are connected by certain bridges. This is the connection. See, these are the connections. These are known as methionine bridges. Here there is a double bond, and here there is a double bond. So actually, this can be written as CH, here double bond, CH, single bond. So this is the connection bridges, which are also known as methionine bridges, which connects the. Now at the center, there is presence of an iron atom. Okay, in heme only the iron is present, so iron is present at the center, connecting the 4 nitrogen atom. Iron is in +2 form. So this is the structure of heme. This is the structure of heme that we are trying to find out. So first we saw there was porphyrin. Over here, pyrrole rings 4 form porphyrin, which add iron to it, we get heme group. Apart from that, there are certain double bonds over here which we need to remember. Over here, one double bond is present. Okay, instead of double bonds on the side, this is just shifting of double bond. Okay, there's double bond here. Similarly, over here is present, and this double bond present over here. So this is the structure we need to remember. Certain things over here additionally we need to remember are the labeling. This is said to be the alpha, beta, okay, gamma and delta carbons, and over here inside it's labeled as 1, 2, 3, 4, 5, 6, 7 and 8. From 1 to 8, if the filling goes, if from 1 to 8 the side groups are methyl, vinyl, methyl, vinyl, then again there is methyl group, then there is propionyl group. Okay, this is propionyl, and then methyl. From 1 to 8 of such group sequences there, then the porphyrin ring which we have got is protoporphyrin. Protoporphyrin which is there is most common one. Moving ahead, we'll go towards heme synthesis. Heme synthesis, for easy remembrance, I just made it see. So that she goes at private UG coaching, poor, and any person whose name starts from H, you can keep it over here. Okay. 7 steps are present over here in heme synthesis, and it occurs in all tissues. Mainly where it does occur is, it mainly occurs in case of normoblast. If I try to use a different color, it mainly occurs in normoblast. Normoblast is a stage of RBC formation, right? See, erythrocytes are said to be formed, and for formation of erythrocytes, first there is hemocytoblast, okay, then there is proerythroblast. Similarly, it goes on, normoblast is present, and then erythrocytes are formed. Okay, so normoblast is a stage of RBC formation. Here, mostly heme synthesis is seen. Apart from that, there is also heme synthesis seen in other tissues, which we'll discuss later on. But it is not seen where? It is not seen in matured RBC. Matured erythrocytes do not contain heme synthesis. Heme synthesis is said to be partly cytosolic and partly mitochondrial. Okay, heme synthesis is partly cytosolic, partly mitochondrial. Now, if we go towards the entire stepwise process, which succinyl-CoA when added to glycine, this takes place in the presence of an enzyme. That enzyme is known as, it is ALA synthase. So this is the enzyme which is going to work. This results in the formation of a product, and that is delta amino levulinic acid. The enzyme used is ALA synthase. Here there is removal of certain things. First, there is removal of coenzyme ASH, and second thing that is removed is CO. Next is this delta amino levulinic acid. This first step which is taking place, it is taking place in the mitochondria. After that, the delta amino levulinic acid moves from the mitochondria to the cytoplasm. In the cytoplasm, delta amino levulinic acid is getting converted to porphobilinogen. This porphobilinogen which is there, it is a monopyrrole. So 4 such porphobilinogen will be forming heme type of structure, right? Monopyrrole means there is only one pyrrole ring, which we had discussed. If there are 4 such, we get heme. So we'll use 4 porphobilinogen in the next step. Now, what is the enzyme used over here? That is important. The enzyme is ALA dehydratase. Dehydratase indicates here there is removal of water. Okay, so this was the enzyme that was used in this step. After that, the next step which is there, 4 porphobilinogen are condensed together with the help of an enzyme which is PBG deaminase. PBG deaminase condenses the 4 porphobilinogen, and this results in the formation. Deaminase means removal of ammonia. 4 ammonia groups are removed. This results in the formation of a structure which is known as HMB, hydroxymethyl-bilane. Bilane is BILANE, okay? Hydroxymethyl-bilane. Now, HMB which is there is undergoing a spontaneous reaction, okay? So HMB which is there, it is undergoing a spontaneous cyclization. The spontaneous cyclization converts it to form a UPG1 structure. UPG1 is now acted upon, okay? UPG1 is acted upon by UPG3 co-synthase. I'm going to convert UPG1 into UPG3. UPG stands for uroporphyrinogen 3. UPG3 which is there is getting converted into another porphyrinogen, that is coproporphyrinogen 3. Now UPG3, we have to remember its structure, or we do not need to remember it exactly, we need to at least know it. The structure of UPG3 is as follows: AP, 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 and here there is PA, where A stands for acetyl group, A stands for acetyl group, and P stands for propionyl group. Now in this step, the enzyme that is used is UPG decarboxylase. UPG decarboxylase. Decarboxylase means CO2 is removed, and amount of CO2 removed are 4. Here what happens is the acetyl group which is present is converted to methyl group. So the structure of UPG which was present is basically only changing in the acetyl to methyl group. I'll just show you over here. So this is CPG. The acetyl group are going to convert into methyl group. Okay, so this is CPG3. Now CPG3 is acted upon by another enzyme to convert it into protoporphyrinogen 3. Here there is removal of 4 hydrogen group to form protoporphyrinogen. What happens here exactly? The enzyme used is CPG. The name this itself is used here as UPG was there, that's why UPG, CPG, CPG. But here the oxidase is used. In oxidase, what happens over here? Basically, oxygen is used and there is removal of CO2. Here hydrogen is not removed because this is oxidase, right? Another thing that happens over here is there is conversion of NADP to NADPH plus H plus. Okay, CPG oxidase is going to convert CPG3 to PPG3. Now this protoporphyrinogen 3, this conversion basically is important because it converts some of the propionyl group to vinyl group. Some propionyl group are converted to vinyl group. So this propionyl group and this propionyl group is converted to vinyl group. Okay, that is nothing but PPG3. Structure of PPG3. After that, the PPG3 is acted upon by another enzyme, that is PPG oxidase. PPG oxidase. This enzyme is going to oxidize PPG to protoporphyrin. Protoporphyrin 3 is going to be converted to heme in this step. Before that, over here, actually what happened was there was removal of 4 hydrogen group, and protoporphyrin to heme, here there is addition of Fe2 plus group. The enzyme used over here is ferrochelatase. It is also known as heme synthase. So this was heme synthesis. After that, we are going to see certain things. Basically, how did the heme synthesis go? This is the mitochondria. This is the cytosol. Okay, let's make a demarcation. Okay, over here first alanine combined with glycine to form ALA. There is not alanine, sorry. Succinyl coenzyme ALA was transferred from mitochondria to cytosol. This ALA was acted upon by dehydratase to form porphobilinogen 3. Porphobilinogen was converted to porphyrinogen. Uroporphyrinogen was converted into protoporphyrinogen. Before that, coproporphyrinogen. Coproporphyrinogen is transported inside, okay? As coproporphyrinogen is transferred inside, wait. This coproporphyrinogen is converted into protoporphyrinogen 3. It is converted into protoporphyrin. This is converted into heme. Now things to notice over here is we are going to look at this regulation. When heme production is increased, it is going to inhibit ALA synthase enzyme. Here the enzyme that is present, ALA synthase. This is inhibited by heme. Over here there are 2 enzymes used, ALA dehydratase. This is inhibited by lead. There is another enzyme over here which is going to convert protoporphyrin into heme, that is ferrochelatase. This is also inhibited by lead. But this is known as feedback inhibition, okay? Heme, which is inhibiting its own synthesis, is feedback inhibition. Over here, when there is transfer of ALA from mitochondria to cytoplasm, there is slow transport. This is also a means of regulation, okay? Slow transport of ALA from mitochondria to cytoplasm is a way of regulation. So we saw a certain way of regulation. Now we'll just write it down. ALA synthase, which is there, inhibited by heme. This is feedback inhibition. Hematin is a structure which is formed when Fe2 plus is converted to Fe3 plus in the heme. The structure formed is hematin. It also inhibits ALA synthase. Compartmentalization of enzyme was the part where ALA, which was formed in the mitochondria, takes time to be transported in the cytoplasm. And drugs and poison effect, we would say barbiturates are a type of drug. They promote it, okay, promote heme synthesis. Then we'll talk about lead. Lead inhibits at 2 points. First was at dehydratase. We saw glucose is also present. It also inhibits ALA synthase. And one thing I need to tell you all that ALA exists in two forms, 1 and 2. ALA1, which is that is present in basically hepatic tissue, and ALA2 in erythroid. Now we'll talk about shunt bilirubin, the topic. When there is, suppose in naturally, heme which is there, it undergoes breakdown, okay, and when heme undergoes breakdown, it forms two things. One is iron and second is biliverdin. Biliverdin further breaks down to give bilirubin. Naturally what happens is heme is present where? It is present in hemoglobin. Hemoglobin is present in RBC. So natural lifespan of RBC is said to be 120 days. So after 120 days, when RBC undergoes destruction, we get bilirubin from it. Okay, this is a natural case. But sometimes what happens is in 10 days itself, the destruction of not RBC, rather heme takes place. And the bilirubin that we obtain then is known as shunt bilirubin. This shows that heme without going inside RBC undergoes destruction. Heme without entering RBC is undergoing destruction to give bilirubin is known as shunt bilirubin.